For this class, the last points we have are food and urine, unknown, ability problem, and the final and the final is like the last study guide.
Yep. Hey, that's a good point. I had to move things around on the website a little bit, and the place where the study guides is shifted on the menu bar. But the study guide that should come up will be one that just basically says there's 2 study guides, and they're for the exam 4 final exam, and they have been revised to correlate with where we broke off for the last exam. So this exam will cover the complement system, inflammation, fever. That was the end of line 2. Line 3, which is the adaptive immune response, and then what we'll cover next week, last week of class, will be the applied diagnostic stuff, the clinical microbiology. So all those study guides are up to date. Okay?
You said there's optional points on.
Well, I've got, they disappeared when they migrated our website. There was a set of videos you can see and answer questions about. So I've got to figure out how I'm going to resurrect those and make them available to you, but I will.
Okay.
Natalie has the videos.
I think that should be automatic option.
You think what?
I think there should be automatic option.
For everyone here today?
No, we don't want optional points. We want bonus.
You want bonus.
Sure.
Greedy, greedy.
Yes.
Hey, we're not greedy.
I am.
She is.
Okay, I get it.
She admits it.
Dude, my feels over.
Yep.
I made it. No.
Okay, let's put together everything we've been working on for the last few weeks. The 2 branches of the adaptive immune system are the cell-mediated immunity and the humoral immunity. And cell-mediated immunity, remember, is directed toward cells that have already been damaged in some way. Maybe they've been infected by a virus or some kind of parasite, or maybe they have become cancerous. Okay? And ultimately what happens if the immune system detects these particular cells is that a series of events occurs that triggers apoptosis in those cells, and the cells self-destruct. Now if you think about it, that's very good if that cell is infected with virus, because if the cell dies before the virus finishes its replication life cycle inside the cell, then that's going to stop that part of the viral infection. Okay? If it's a cancerous cell, then if that cell dies before it can divide and form new cells, then that can arrest tumor growth. In reality, we probably all have cancer cells developing in our body, not infrequently, okay, because mutation occurs. But the immune system is pretty good at recognizing those cancer cells. And one of the things that's a big area of cancer research is why in some cases do cancer cells escape detection by the immune system? And are there things we can do in cancer therapy to enhance immune system function? That is a much better treatment from the patient's standpoint than a blanket shotgun chemotherapy approach, which does kill cancer cells but kills a lot of healthy cells as well, and consequently has those side effects associated with chemotherapy. So if we can enhance immune function, then we're going to enhance the action of cells that are targeting those cancer cells specifically. So there's a lot of relationship between immunology and cancer study. Okay, the major players in the adaptive immune response are antigen-presenting cells. And what are the major antigen-presenting cells? Macrophages. What else? Dendritic cells, kind of their cousins. And then B lymphocytes can also be antigen-presenting cells. Which MHCs do these cells have? They have two. Do they have ones? Yes, they do. Remember, all nucleated cells have ones. Okay. Another important player in this response is the B lymphocyte. The B lymphocyte can play kind of a number of roles in the immune response. It can be an antigen-presenting cell, but what is its major function? What's the major thing it does? Secretes antibody. Okay. And then the other set of cells that are major players in this are the T cells. And we're going to talk primarily about three types of T cells: T helper 1s, T helper 2s, and T cytotoxic cells. So what are these guys going to do? Well, we set up here. They're going to kill damaged cells. What are T helper 1 and T helper 2s going to do? They're going to provide some kind of assistance. Okay? T helper 1s assist the T killer cells or the T cytotoxic cells. T helper 2s assist the B lymphocytes and antibody secretion. So those helper cells play a really central role in this whole immune response. Okay, so we already talked about this. T helper cells, cell-mediated immunity involves T killer cells destroying or inducing the destruction of damaged cells. Here's our major players that I just listed, and referencing their CD receptors on a couple of the diagrams you're going to see. You're going to see the important role that CD receptors play in the immune response. And then this talks about some of the important functions of those. Okay? So T helper 1 cells can activate T killer cells. They can secrete a lot of cytokines. T helper cells primarily drive B cell proliferation, and they involve some different cytokines that they secrete. And then the T cytotoxic cells kill those virus-infected cells. Cancer cells also play an important role in graft rejection. So let's talk about the cell-mediated immune response first. Okay, the involvement of the T cytotoxic cells. These are the primary events that take place in the sequence that results in activation and action by the T cytotoxic cells. A big part of it is going to involve antigen presentation to both a T helper 1 cell and a T killer cell. So that antigen presenting cell is actually going to be showing engulfed antigen to two kinds of cells, the helper 1 and the cytotoxic. That's going to result in activation of both of those cells. So T helper 1 cells are going to start dividing. And as they produce cytokines, that's going to help to activate T cytotoxic cells to start dividing. We then get clonal expansion. 1 cell gives 2, 2 cells get 4, 4 cells get 8. And one of the interesting things that happens during this clonal expansion is the T cytotoxic cells start producing their own cytokines, which further enhance cell division. They kind of self-stimulate themselves. And so we've got a very interesting positive feedback process going on here, which results in this huge population of cells developing to deal with the threat. Remember, a T cytotoxic cell at one time can only kill one infected cell. If you've got a whole bunch of epithelial cells that were infected by a cold virus, for example, then that takes a whole lot of T cytotoxic cells to bind to those cells and then signal them to die. Now, can a T cytotoxic cell move on to a different cell once it's activated, killing it once? Yes. But you still need a whole bunch of T cytotoxic cells to deal with all of that infected tissue. Okay. Here's a diagram you really should have. If you're not in the habit of printing out PowerPoint slides, this is one that you should print out because this really gives the summary of activation of T killer cells. So here's our antigen-presenting cell. This is a dendritic cell. This has encountered antigen someplace in the body, maybe in the skin, for example. Okay? Dendritic cells, remember, have toll-like receptors on their surface. And toll-like receptors are looking for PAMs. Remember that acronym? Pathogen-associated molecular patterns. So these are molecules that are traditionally associated with a pathogen. If the toll-like receptor binds to a PAM, then that activates that cell to then phagocytize. And then this particular cell, once it's phagocytized, presents antigen on its surface. Now, it can actually present that antigen in both a class 1 MHC and a class 2 MHC. And here's why. When it engulfs the antigen, remember it's taken into a phagolysosome, and then vacuoles in the cytoplasm that have MHC2s on the inside of that vacuole fuse with that phagolysosome. And then those MHC2s with the broken down antigen are put on the cell surface. Okay, so that's what's shown right over here. But some of that pathogen accumulates just in the regular cytoplasm of the cell, like any cellular protein. And remember we said that cellular proteins are constantly recycled in the cell and broken down. And if they are, then there are MHC1 receptors on ER, and they bind to these pieces of broken down protein, including some of the pathogen, and then they will move out to the cell surface and present cell proteins in general, including pathogens, on the surface of the cell in the context of an MHC1. Now, this is something important. T cells, we say, are MHC restricted. That means that they can only see an antigen if it is displayed along with an MHC receptor. Okay? So this epitope here has to be nestled into this particular MHC1 molecule or an MHC2 over here. Otherwise, a T cell cannot see it. It's as if it doesn't exist. Okay? T helper cells are class 2 restricted. They can only see an antigen in the context of a class 2 MHC. T cytotoxic cells are class 1 restricted. So we got this crazy dendritic cell that's carrying around these badges, if you like, of epitopes displayed on MHC 1s and 2. Where do they go? They migrate away from the skin and head toward the lymph nodes, because that's where those subpopulations of educated T helper cells and T cytotoxic cells are sitting there waiting. Okay? And remember, they have a specific receptor for a particular antigen. So basically, the dendritic cell starts crawling around through the lymph nodes and showing what it's got to all those T helper cells and all those T cytotoxic cells. I don't know how they calculated this, but they believe that a given dendritic cell could potentially move through every lymph node in the body and show what it has on its surface to all of the T cells once a day. Now, it's going to stop if it encounters its partner T cell. But theoretically, it could move through all the lymph nodes. That's pretty impressive. Okay?
Are there smooth muscles that aid the flow?
No, they're amoeboid movement.
So just
Yep, pseudopodium. Yep. Now, I mean, they could get into, I suppose, lymphatic fluid, and as they're pushed through the lymph by muscle contraction, that would move it faster. Okay. Okay, so here is our dendritic cell presenting an epitope to a T helper cell. Okay, that T helper cell is a particular receptor that matches the epitope. The T helper cell is restricted, so it can only see the epitope in the context of this MHC2. It's there, they can see it. Okay. When those two bind, some chemical signaling occurs, and the dendritic cell begins to produce this cytokine, interleukin 12. That provides the second signal. This binding of the epitope was the first signal that begins to activate the T helper cell. This particular T helper cell turns into a T helper 1, and it starts secreting its own cytokine, interleukin 2. Okay, simultaneously, the inactivated T cytotoxic cells are showing the epitope in the context of an MHC1 receptor. Just remember, they're class 1 restricted. They can't see this antigen as part of this class 2. It's got to be part of the class 1. When this presentation takes place, that induces these inactive cells to start producing receptors for interleukin 2. They're getting ready to be stimulated by this interleukin 2 that's being produced by this T helper 1. When interleukin 2 binds to those receptors, that starts clonal expansion. And we get memory cells, and then we get activated T killer cells, and they basically then start stimulating themselves. They start producing their own interleukin 2, which induces more interleukin 2 receptors to develop on the surface. So these just start producing interleukin 2 like crazy. And that causes cells to divide really fast, and then these cells produce interleukin 2. So these are now the primed and activated T cytotoxic cells. And notice that they've received a number of signals before they've been activated. They've received the signal of seeing their antigen presented to them, and then they've received the signals of interleukin 2. Okay. Now these are ready to go. So these activated T killer cells now can leave the lymph nodes and start moving throughout the body and looking for virally infected cells or cancer cells which bear the same epitope which caused them to be activated. Okay? So what happens? When a virally infected cell displaying a virus antigen on its class 1 MHC matches up with an activated T killer cell, and this shows you some of the receptors that match up in order for that to happen, then some interesting things begin to happen within the cytoplasm of that T killer cell. Some molecules start to be packaged up in vesicles. Those are proteins called perforins. And those vesicles fuse with the membrane, are released over onto the cell that's infected. This right here is sometimes called the immunological synapse, kind of like the neuron synapse. Okay? And those molecules then polymerize on the surface of the cell and basically form a pore complex in the cell. What's that look a lot like what we've seen before? It's like complement, okay? The attack complex, complement, okay? But then they've got another killing mechanism. And they package up some different proteins that are enzymes called granzyme. And those vesicles are released to the surface of the cell. And the granzyme moves across the immunological complex and starts flowing through this perforin pore complex. What does that do? Well, in the cytoplasm of normal cells, including your cells, are some inactivated suicide enzymes or apoptotic enzymes. And in the presence of granzyme, they become activated. So now they're active. And that induces the apoptosis process, and the cell shrivels and disintegrates and dies. Once this has happened, this T cytotoxic cell could then move on and find another viral infected cell. And another. And obviously, the more of these cells that there are, then the more efficient it's going to be searching out all of the viral infected cells within the body. So that's why this clonal expansion is so important, to generate a big population of those cells that can go around the body and kill your cells that are causing problems. Okay? So that's the T cytotoxic cell. But again, look at the complex interplay that takes place between antigen-presenting cells, helper cells, cytotoxic cells, and the role that these interleukins play. It's really, really critical that this system be set up so that it takes multiple signals to activate it, because it's a destructive system. Okay? It is really the nuclear launch against your body's tissues. And we don't want to just have a single signal, that activates this destructive system. Okay? So it's not just enough to see the foreign antigen on the surface of an antigen-presenting cell. We want to make sure that we get the proper set of signals. And they've got to occur in the proper sequences as well. It's kind of like a combination lock. Okay? I can tell you a combination lock can be a combination of 34, 2, 8. But if you didn't get the sequence right, it wouldn't open. So these interleukins also have to be produced, but in the proper order. So it's a very orchestrated process, and when it all comes to fruition, then we get this very impressive killing process that's occurred. So any questions about the T cytotoxic response? Okay, humoral immunity. Humoral immunity results in production of antibody. And we talked about all of the different ways that producing antibody can help you out. Neutralizing virus so they can infect cells, activating complement, serving as opsonins, agglutinating pathogens, precipitating toxins. Okay? And again, there's going to be a pretty complex interplay between antigen-presenting cells, T helpers, and B cells this time. Now, when we talked about antigens, we said that there were two categories of antigens. There are T-independent antigens, and there are T-dependent antigens. T-independent can stimulate B cells directly without the helpers being involved. This is a fairly limited response. It results in response to molecules that have repeating subunits, carbohydrates, lipids. And the significance of the repeating subunits are that they can cross-link B cell receptors. That's a process called capping. And then that cross-linked molecule can be pulled into the cytoplasm of the cell, which is the main event that initiates then that B cell to start producing antibody. But we only produce IgM. Remember, IgM is normally the antibody that's produced first. But then a phenomena occurs in most cells in response to most antigens, but not T-independent, that results in IgG or IgA or IgE being produced later. What do we call that phenomenon? You can produce a different time. Class switching, okay? So we don't get class switching occurring in response to T-independent antigens. Why not? Because we don't get the right cytokine signals because T helpers are not involved. So, here's an educated B lymphocyte, probably sitting in a lymph node someplace. And it has a particular B cell receptor that it's built out of those blocks of genes. And now it is committed to only making that receptor and then making an antibody with the same specificity of that receptor. And as it turns out, only one class of that antibody. Okay? It makes the M and it never switches over to make other classes of antibody because it doesn't get the proper signal. So here's our polysaccharide with repeating subunits. All of these little subunits are epitopes. And so the multiple B cell receptors bind to this. That pulls the receptors together. Endocytosis occurs. And that basically signals this plasma cell to now become activated and start producing IgM. Very little memory response associated with this. You can get some memory response, but not much of a memory response associated with this. Fortunately, T-independent antigens are kind of in the minority. Most of the antigens that are going to cause you problems are the T-dependent variety. Okay, I like this slide. It's a little confusing first when you look at it, but this shows you kind of locations within the immune system where things are going on. And so this side over here is talking about the T-independent antigen response. So what it's showing you here is what's happening in the bone marrow to result in those educated lymphocytes. And then those educated lymphocytes can move into a variety of locations, the nodes, the spleen. Most of the T-independent responses occur within the spleen. So they see these particular repeating subunit antigens, and that causes this capping to occur, and then these cells differentiate into plasma-producing cells, which make IgM. I'll come back to this in a minute, but this is kind of the T-dependent antigen response, which happens more frequently in the lymph nodes. Okay, so T-dependent antigens. They require the help of T helper cells. They also require the involvement of dendritic cells. Those are the antigen-presenting cells. And again, a minimum of 2 signals are required before T-independent antigens can start a B lymphocyte to produce antibody. One is recognizing your epitope. That's kind of a no-brainer. But then the other is cytokines that are produced by T helper cells. These are the dendritic cells. I don't think I've ever shown you this particular picture, but these are those macrophage cousins that play such an important role as antigen presenters. And they have these little dendrites or extensions coming off of their cytoplasm. That's why they're called dendritic cells. Okay, here's the second diagram you should have. These are wonderful reviews for this part of the exam. Okay? So, what have we got going here? This is a T-independent antibody response. And here is a population, probably in a lymph node, of a whole bunch of different T helper cells. Each of them has a different B cell receptor. Okay? They develop their own B cell receptor by that gene shuffling process. They're sitting there waiting. Here's an antigen presenting cell, a dendritic cell, let's say. Okay? Remember, T helper cells are class 2 restricted. So they can only see this epitope, the foreign antigen that the APC is ingested, in the context of that class 2. And this shows you how some of the receptors particularly the CD4 on the T helper cell, interact with the MHC2. So all these receptors have to match up before we get proper dot, if you will. Okay? So now we have T helper cells. And under the influence of interleukin 4, they differentiate into a T helper 2 cell. Remember, T helper 1s are involved in the T cytotoxic response. So interleukin 4 here causes T helper cell 2s to develop. Okay? Here's a population of B lymphocytes, each with their own receptor. And when the proper B lymphocyte with the proper receptor sees the proper T helper cell with the antigen on its surface, and that docking happens, that then results in production of more interleukin 4, which activates the T helper cell now to divide and form plasma cells and memory cells this time. And these start cranking out antibodies, but then additional interleukins can be produced by T helpers and by some of the macrophages that cause class switching to occur. So in addition to producing the IgMs, you can see those right here, the big ones, these can produce IgG or IgE or IgA. So we get those different classes of antibody produced in response to the T-dependent antigen. So now we start cranking out antibody, and the antibody does its thing. So this then just kind of puts it all together now, showing the T-independent antigen side over here happening in the spleen, and the T-dependent antigen process happening over here in primarily the lymph node. I was looking at the animations that are on that website that I sent you for the textbook, and there's some pretty good animations on there for the immunology section that kind of break this down into various digestible bites and show you some of the kind of interactions and animation. And I think some of those could be helpful, just for visualizing them. So you might want to be aware of those. And then this is just kind of a summary slide that kind of just puts this all together in one slide. I'm not going to go through that all right now. Here's just another summary, kind of as a flow diagram. And so these particular, and then this kind of shows you the whole post-immune response, levels 1, levels 2, levels 3. And so I think some of these last slides are really good summaries for kind of putting this whole thing together. Now, there's a couple of additional considerations that I would just like to throw in at the very end here about the adaptive immune response. HIV is so destructive to the total immune response because HIV binds to CD4. That's how it gains entrance into those cells. And CD4 cells are T helper cells. So HIV knocks out your T helper cells. And T helper cells play an important role in both the cell-mediated immunity and in the humoral immunity as well. So that's why they are so devastating in terms of compromising your overall immune system. I've mentioned this already, but I'll mention it again. Steroid compounds, including cortisols and other sterols, tend to function as dramatic depressors of the immune system. We mentioned that cortisols were given to transplant patients to suppress their rejection response. When the human body is under stress, it tends to produce a lot of cortisols. And that's why many times illness follows periods of prolonged stress. It's not uncommon for students to get ill immediately after finals. Taking out a lot of sterols, and your immune system is really depressed at that particular point. exercise is good for your immune system. When your body responds to exercise, one of the things that it does is it starts producing some compounds called endorphins and enkephalins. And these are basically natural opiates. I've never experienced it before, but supposedly if you run a lot, for example, you really can get into kind of a runner's high due to the production of these enkephalins and endorphins. To me, running has always been just a huge chore. Never gotten past that point. But these particular compounds can also be stimulators of the immune response. And they really do seem to enhance immune function. A pretty fascinating cross-discipline that's developed in the last decade or so is the discipline of neuroimmunology. And this really studies the relationship between the nervous system and the immune system. And what we're finding out now is that there is a lot of chemical communication that can occur between neurons and immune cells via cytokines. You've all heard stories before about how somebody has basically, through will, beaten a disease like cancer. There is actually some biochemical basis for that kind of response. And that there is kind of talking that can occur between nerve cells and immune cells. And your state of mind can really influence the status of your immune system. People who are depressed don't get as much nervous stimulation of their immune cells as people who are not depressed. So this whole idea of the interconnectedness of those systems is a really fertile area of investigation right now. Used to think that they were pretty separate systems. There really wasn't much crosstalk between them. And now we're finding out that that's not the case at all. Well, we've just scratched the very, very surface of immunology. We could easily have a whole semester immunology course and still not even get into that much depth. But hopefully you have some kind of an idea of the cooperation of these particular cells that takes place. And what a remarkable system this is. One of the things that really fascinates me is, how did this system evolve? How did all this cooperation among the cells evolve, and all this chemical communication, this restriction of only certain cells responding to other cells? It's really pretty fascinating. The last topic that we're going to talk about after we get back are applications of immunology, and particularly using antibodies in testing for disease. So we're going to talk about some serological tests using blood serum to do tests for disease. And that will take up the last 2 or 3 periods of class. So that's that one final chapter in your book. So between that and working on your unknown, that's going to be your responsibility. I'm getting really soft here. I gave you 5 minutes the other day, 10 minutes off today.
Oh, yeah.
So, have a good Thanksgiving, everybody. And come back ready to go for your last week. Check with
Is that what she's talking about, like,
I know she's in neurobiology. I don't know if she's in neuroimmunology.
okay.
Yeah.
Gotcha.
And then, I have.
Yeah.
I think I have pictures of everything.
Just to sort out what's what.
I still bet you